Let's talk about passive and active transport and why each is important. So the cell membrane is an important part of each cell, providing structure to each cell and protecting each cell from its outside environment. Because of their phospholipid bilayer, the cell membrane can determine what can or can't enter the cell, which is key to upholding the balance of homeostasis. So the way they do it is semi-permeability. So what is semi-permeability? So as we know, cells all work best in different environments, whether it's in an environment with higher pH concentrations or in areas with higher solvents outside the membrane, like sodium. If our cells can control what goes in or out of the cell, it would disrupt the homeostasis inside the cells, exposing the cytosolic contents. Luckily, our cells are semi-permeable, allowing certain molecules to enter while having others stay out, allowing the cell to remain homogeneous despite the different concentrations. So some molecules can enter the membrane really easily just by drifting in and out, while others must use energy. So how do we actually classify the movement within the cell membrane? And it's actually really easy to remember, with passive requiring no energy and active requiring energy. Think of it as the membrane has to activate to accept these molecules. So let's talk about passive, and it's generally with diffusion. So what is diffusion? The simplest way to state it is the movement of particles down each gradient, a gradient being any imbalance in concentration, thus allowing the particles to be distributed evenly. The molecule that is most likely to go through simple diffusion is water, but it goes by a different name, one that we may be familiar with, known as osmosis. So simple diffusion is actually really synonymous with its name. The molecules must move down their gradients through the membrane. Molecules that practice simple diffusion must be small and nonpolar as well. Facilitated diffusion is diffusion that is helped along or facilitated by a membrane transport channel. These channels are generally glycoproteins or proteins with carbohydrates attached that allow molecules to pass through the membrane. These channels are almost always specific for either a certain molecule or a certain type of molecule, such as an ion channel, and they are tightly linked to certain physiologic functions. For example, there is a transport channel known as GLUT4, and it's important in diabetes because GLUT4 is a glucose transporter found in fat and skeletal muscle. Insulin triggers the GLUT4 to insert into the membrane of these cells so that glucose can be taken in from the blood. Since this is a passive mechanism, the amount of sugar entering our cells is proportional to how much sugar is consumed, up to the point that all the channels are being used. Okay, so active transport. Sometimes the body needs to move molecules against their gradients. In that case, it needs energy to actually move those molecules. There are two types of active transports, known as primary active transport and secondary active transport. Transport that uses ATP is known as primary active transport, but what does it actually look like? Let's use the sodium-potassium pump that helps maintain resting potential in the cell. This protein uses the energy released from the hydrolysis of ATP to create 3 sodium ions and 2 potassium ions. Secondary active transport moves multiple molecules across the membrane, powering the outward movement of one molecule, also known as the one going against the gradients, with inward movement of the other molecule, known as the evening out of molecules. Since both molecules moved in the same direction, this molecule is known as a symporter. Proteins that allow molecules to go in opposite directions are antiporters. So what about bulk transport? So sometimes the particles are too big for cells, so they go through bulk transport. Large particles or large quantities of smaller particles are then moved across the cell membrane. It happens by enclosing the substance to be transported in their own small bits of membrane, which can then fuse with the membrane of the cell to move across the substance. And is known as endocytosis. Endocytosis is a general form for the various types of active transport that move particles into a cell by enclosing them in vesicles made out of a plasma membrane. There are variations of endocytosis, but follow the same basic process. First, the plasma membrane of the cell folds inwards, forming a pocket around the target particle or particles. The pocket then pinches off, leaving the particle trapped in a newly created vesicle or vacuole inside the cell. Endocytosis is actually made up of, can be categorized as multiple things such as phagocytosis, pinocytosis, and receptor-mediated cytosis, or endocytosis. So phagocytosis is the method of transport by so-called eating a cell. By that, I mean it is a form of endocytosis that envelops large particles like cells or cell debris, so they're engulfing it. Once the cell has successfully engulfed the target particle, the pocket containing the particle will pinch off from the membrane, forming a membrane-bound compartment called the vacuole. This vacuole will later fuse with lysosomes, which have enzymes that break down the engulfed particle down into its basic compartments like amino acids and sugars, which can be used by the cell. Pinocytosis is the method in which cells take in extraneous liquids while enveloping. They occur quite frequently and are much smaller than phagocytosis. The last form of endocytosis is receptor-mediated endocytosis, which is a form of endocytosis in which receptor proteins on the cell's surface are used to capture a specific target molecule. The receptors, which are transmembrane proteins, cluster in regions of the plasma membrane known as coated pits. When the receptor binds to their specific target molecule, endocytosis happens and the receptors and their attached molecules are taken into the cell in a vesicle. The coat protein participates in the process by giving the vesicle its round shape and helping it bud off from the membrane. Although receptor-mediated endocytosis is intended to bring useful substances into the cell, it may sometimes allow less friendly particles to gain entry by the same route, such as viruses. So exocytosis is a form of bulk transport in which materials are transported from the inside to the outside of the cell in membrane-bound vesicles that fuse with plasma membrane. Some of these vesicles come in from the Golgi apparatus and contain proteins that are made specifically by the cell for to be released outside, such as signaling molecules. Other vesicles contain waste that the cell needs to dispose of, such as the leftovers that remain after phagocytosis has occurred. These vesicles are transported to the edge of the cell where they can fuse with the plasma membrane and release contents into their extracellular space.